Hi guys, Dr. Alia here. This is a short introduction for our next three chapters concerning DNA replication, transcription, and also translation. So all these three chapters are basically just talking about how from DNA we get protein. First off, as we know, DNA is the genetic material that defines every cell. So before a cell duplicates and is divided into new daughter cells through either mitosis or meiosis, so other things, biomolecules, organelles, and also the DNA must be copied to be distributed among the cells. So DNA, which is found within the nucleus, must be replicated in order to ensure that each new cell receives the correct number of chromosomes. The process of DNA duplication is called DNA replication. Replication follows several steps that involve multiple proteins called replication enzymes. You'll learn about these later, and RNA. In eukaryotic cells, such as animal cells and plant cells, DNA replication occurs in the S phase of interphase during the cell cycle. So the process of DNA replication is vital for cell growth, repair, and reproduction in organisms. The central dogma of biology is the process by which the instructions in the DNA are converted into a functional product. It was first proposed by Francis Crick in 1958, one of the people who discovered the structure of DNA. Remember him? Okay, so the central dogma explains the flow of the genetic information from DNA into RNA and then into a functional product, the protein. The central dogma suggests that DNA contains all the information needed to make all of the proteins, and RNA is a messenger that carries this information to the ribosomes. And then the ribosomes serve as factories in the cell where the information is then translated from a code into a functional product. The process by which the DNA instructions are converted into a functional product is called gene expression. Gene expression has two main stages, transcription and translation. In transcription, the information in the DNA of every cell is converted into small portable RNA messages here. And then during translation, these messages travel from where the DNA is in the cell nucleus out into the ribosomes, where they are read to make specific proteins. Here, the proteins. So this is translation. The central dogma states that the pattern of information that occurs most frequently in our cell is first from existing DNA to make new DNA, which is DNA replication, from DNA to make new RNA, which is transcription, and from RNA to make new proteins, which is translation. Next up, we need to understand the genetic code. The genetic code is a sequence of nucleotides in DNA and RNA that determines the amino acid sequence of proteins. So, although the sequences of nucleotides in DNA carries the information for the protein sequences, it is not made directly. Remember, it has to be transcribed into a messenger RNA first. And then from there, it directs the information into final product, the protein. So RNA is composed of 4 nucleotides. Remember this. You have your adenine, your guanine, cytosine, and then the uracil, and three three adjacent nucleotides constitute one unit called the codon. And one codon codes for one amino acid. For example, if you have AUG, is a codon that specifies the amino acid uh, methionine. And then there are actually 64 possible codons, 3 of which do not code for amino acids but actually indicate the end of a protein. So there's the certain codes. The remaining 61 codons specify the 20 amino acids that make up proteins. How to read this genetic code? Of course, there is a table. Like you can refer your lecture notes for the table of codons. So there just an overview of the chapters that we are going to go into. With the video assignments that I have given you, you will get a deeper understanding. You will be going deeper into the mechanisms involved in DNA replication, transcription, and also translation. So keep calm and make your videos. That's all for today. Bye.